is entitled Inflammatory Keloid Fibrolasts Promote CCL2-dependent monocyte recruitment and polarization toward M2-like macrophages. I won't describe a keloid disorder, but I want just to maybe to precise some keywords: chronic inflammation, mechanical cues, genetic susceptibility, which were all addressed this morning. And regarding the complexity of the cell and tissue microenvironment of the keloid disorder, and the fact that the keloid overgrows over time beyond the wound measures, we believe that we can maybe find and act on something at the periphery of the keloid, where the mechanics is very complex and where the inflammation is very high. Because we believe that we have to work on the vicious circle which takes place at this location, where the mechanical microenvironment plus TGF-beta influence the proliferation, differentiation, ECM deposition, and so on regarding fibroblast physiology, which in turns recruit more inflammatory cells, and especially type 2 macrophages, which also enhance collagen deposition and maintain the high level of fibrosis, which induce non-resolution of the inflammation, and so on and so on. So the question we have in mind is, what's the inflammatory role of keloid fibroblasts? This question is based on previous work, which have been presented in Montpellier during my last participation at the Keloid Symposium. And we worked with Dr. Zélie Diran, my former PhD student, and Sylvain Perruche, a colleague from the university. We worked on the keloid fibroblast fibrogenic phenotype, and we have shown that this fibrogenic phenotype and its regulation is made by the macrophages, of course, but also by macrophages polarization. And we get some tissue from the Besançon Hospital, and we have confirmed that there's an overexpression of type 2 macrophages markers inside the keloid tissue. And we asked the question, and the question was if the type 2 macrophages could be one of a major contributor of keloid pathogenesis, in accordance, of course, with the description we are able to find in the literature. So we polarized by ourselves some monocyte in the lab into type 1 and type 2 macrophages. And we used the secretoma of these polarized macrophages to treat keloid fibroblasts in order to experience the fact that if M1 or M2 are able to differentially modify keloid fibroblasts regarding the migration, proliferation, myofibroblast differentiation, ECM synthesis, and matrix remodeling. So I don't want to dive into all the results, but our conclusion was this one. Regarding the effect of M1 macrophages secretoma on keloid fibroblasts, we have shown that this secretoma, M1 secretoma, was able to reduce migratory capacity of keloid fibroblasts in the presence of TGF-beta. The secretoma was also able to prevent the TGF-beta induced increase in type 1 collagen production and accumulation by keloid fibroblasts. And we have shown that M1 macrophage secretome was able to induce and to make fibroblasts evolve to a fibrolytic phenotype. It was not the case for M2 macrophages secretome, because we have shown that this kind of cell soup was able to increase migration of keloid fibroblasts in response to TGF-beta in a synergistic way, was able to enhance chemotactic recruitment of keloid fibroblasts. The M2 macrophage secretome was able to induce the myofibroblast differentiation, and again with TGF-beta in a higher way, and also increase the ECM remodeling capacity of keloid fibroblasts in 3D collagen gel models. These data were published 2 years ago with the contribution of Dorothy. Thank you very much, Dorothy. And this paper is for me a proof that I was so right to contact Michael several years ago to join the Keloid Symposium, because this is an example of what we can do together. And I'm very proud of this publication together with Dorothy. Thank you again. We are waiting for the next one. And maybe the next one is based on this new data, because the result I exposed to you was in the way from the macrophages to fibroblasts in the crosstalk between the two type of cells. So our question now is, what about the effect of keloid fibroblasts on monocyte / macrophages M1 or M2? So we went back to our nitrogen tank to defreeze some cells and to perform a kind of bulk RNA sequencing on keloid fibroblasts. This is the work of my PhD student, Yasmin, and she compared the transcriptomic profile of 3 primary keloid fibroblast cells compared to I don't want to say normal fibroblasts, let's say non-keloid fibroblasts from patients who are not prone to develop keloid. It's more difficult to say, but difficult to say normal. And of course, we have this result showing that we have 2 different population, keloid fibroblasts and normal fibroblasts. And regarding all the analysis we perform with this data collection, we have shown that we have a different pathway which are upregulated, and one of these pathways is the chemokine signaling pathway, which was very interesting regarding the question we have in mind. More specifically, I won't dive deeply in all the differences, just to focus on some cytokines, and one of the most important for us is CCL2. So you have here the data from the RNA-seq, and as we can see on the slide, regarding this transcriptomic profile, CCL2 is very increased in keloid fibroblasts. It was shown this morning that CCL2 is known to be upregulated keloid, but we have the proof that it's also the case at the cellular level, especially in keloid fibroblast. This result from the RNA sequencing was confirmed by our own PCR data at the protein level too. I will give some result just after. I just want to remind you with CCL2 effects, and the old name of CCL2 for the younger ones was MCP1. And CCL2 is a critical pro-inflammatory chemokine that acts as a major driver of fibrosis across multiple organs, including skin and especially keloid, and is able to promote fibrotic diseases by recruiting immune cells, monocyte and macrophages, to the site of tissue injury. So briefly, CCL2 is able to drive fibrosis regarding immune cells recruitment, of course, but also myofibroblast activation and angiogenesis. And as I said before, the CCL2-CCR2 axis is well described in several reviews, several publications. You can find easily on PubMed regarding hepatic fibrosis, intestinal fibrosis, cardiovascular, pulmonary, renal fibrosis, and so on. And not so much review or a paper regarding CCL2-CCR2 in keloid. That's why we dive into this point and try to look at the in vitro level, at the cell level, about CCL2 in keloid. 2 examples of data you can find on PubMed, data which support our question. We beyond CCL2, we perform multiplex analysis at the protein level, and we prove that fibroblasts, from the secretome point of view, are considered as inflammatory and pro-inflammatory cells with a rich chemokine environment. And again, at the protein level, we find a high upregulation of CCL2 protein expression in keloid fibroblasts compared to non-keloid fibroblasts. Because we've got some money in our pocket, we about some multiplex membrane to perform proteomic profiling of keloid fibroblasts regarding ECM degradation profile. And compared to normal fibroblasts, you can see easily here on the graph that the major protease are downregulated in our cells compared to normal ones in favor of course, ECM protein non-degradation and accumulation. We have cells in our incubator, so we have decided to perform cytometric analysis regarding some special markers given from colleagues who work on the CAF, cancer-associated fibroblasts, because as it was said this morning, keloid disorders can be looked at by the prism of cancer markers, or let's say that keloid shares some similarities with cancer. That's why because we had some colleagues working on CAF, why don't use their marker on our keloid fibroblast. So we have done that, and surprisingly, we observed a high upregulation of podoplanine expression in our keloid fibroblasts compared to normal ones. And podoplanine is one marker in cancer, and CAF of cell invasion is a CAF markers. So maybe that podoplanine can also be considered as an invasiveness markers in keloid fibroblasts, which are not only ECM overexpression cells. If you go back to CCL2 and the hypothesis that keloid fibroblasts compared to normal ones could be able to recruit more monocytes and macrophages than in a physiological situation. So as I said before, we confirmed all the data about CCL2 at the RNA level and the protein level, and also by immunostaining, as you can see on the picture. And we also use the secretoma from keloid fibroblasts to address the question of chemoattraction of monocyte. So we perform a THP1 migration model with Boyden chamber in a Incucyte S3 microscope, which is a real-time microscope. And we were able to follow over time the attraction of THP1 across a membrane according to the secretoma we had to the well, either keloid fibroblast secretoma or normal ones. And we have shown that the secretoma from keloid fibroblasts was able to chemoattract a higher number of THP1 than in normal situation. I don't show the extra data we obtain here, but we have confirmed that this higher chemoattraction was related specifically to CCL2. We have used some blocking agent and antibodies and so on to prove that it was the fault of CCL2. So we are now in a situation where we have a lot of proof to maybe to prove our mechanistic hypothesis, which is the following one: keloid fibroblasts, which can produce high amounts of CCL2, which can be maybe considered as biological markers. So the high expression of CCL2 could lead to an aberrant recruitment of monocyte at the wound site, or let's say at the keloid site, which after recruitment can be polarized into M2 type macrophages that persist at the wound side, at the fibrotic tissue. And as we work on before, the M2 macrophages can enhance the fibrogenicity of keloid fibroblasts. All these things fueling the vicious circles of inflammation, fibrosis, and keloid tissue invasion over the normal or the non-keloidian skin. And the question now could be if the CCL2-CCR2 axis can be looked as a target for keloid treatment or recurrence prevention. So our ongoing research and future directions. So with Yasmin, we are going to fully demonstrate the in vitro implication of CCL2 in monocyte recruitment from keloid fibroblasts. We also work on the determination of the impact of the inflammatory microenvironment on CCL2 production by keloid fibroblasts. We want also to evaluate the impact of keloid fibroblast secretoma on macrophages polarization in hypoxia. We have some data not shown here in favor of M2 polarization in such situation. And regarding the secretoma we used, the full secretoma, we want to know if the biological effect we have shown is related to the soluble factors or the EVs, the extra vesicle, of course, in order to better characterize the phenotype and the mechanical properties of our pathological fibroblast. So, of course, I thank all of my colleagues of the Université Marie and Louis Pasteur, all of you for your attention and your question, especially Dorothy and Michael and Frank for their friendship and their warm welcome today and collaboration. Thank you so much.
Are there any questions? Can I ask a question? When you combine or keloid tissue and you do research on normal skin, in this research, you showed that the normal skin was from the same patient?
Unfortunately not, because the keloid fibroblasts we use come from tissue after earlobe keloid resection. And our surgeon colleague perform intrakeloidian resection. That's why we don't have any non-keloidian tissue from the same patient.
Okay.
So we use the abdominoplasty.
I understand.
As control is not, of course, the best controls as possible, but we have only this kind of tissue.
So it is possible that you look at different fibroblast types between different people, because the one is developing keloid and the other is not developing keloid.
Yeah, of course, of course.
So what we did, we tried to excise a keloid with a piece of skin adjacent to it and look at the skin nearby the keloid and farther away, because we showed that the normal skin nearby the keloid is different in expression of interleukins already. So be careful, and we have to be careful and take in mind that, but to try to get normal skin of a keloid farmer, that's of course difficult, because maybe they will develop a keloid as well.
Yeah, sure.
So, but we have to keep in mind that maybe we are looking at different things.
Yeah. And the other, if I can complete your comment, the other problem we have is regarding the monocyte we used and the macrophages we polarized, because the monocyte did not come from the keloidian patient.
Yeah.
It was also a difficulty for us.
Yeah, I understand.
Yeah.
Okay. One more question.
Yeah, on a related note, since your keloids and your normal samples were from different patients, but do you normally see, if you've done any more transcriptomic, do you normally see the keloid patient samples cluster closer together?
Yeah, yeah. And beyond that, if we look at the literature, because there are more and more RNA sequencing, either bulk or single cell ones in the literature, our little study is in accordance with such data. So, of course, we have to increase the number of cells we sequence and try to maybe develop our own single cell strategy to increase the power of our conclusion.
Thank you.
Let's go for the next talk.
Thank you so much.
Thanks.